So I'm going to talk about some more recent periods. I'm going to talk specifically about food and the foods that we eat. Food is something that unites all of us. It's something we all consume. It's something that we share in common. It delights us, it excites us, it nourishes us, and it sustains us. But I think one question I often think to myself is, well, how did we come to eat like this? Following on Zeray's talk, how did we come to eat out of this modern supermarket? And as an archaeologist who researches ancient diets, I often get asked, what should we eat? What are we evolved to eat? And this is a really important question, but I have to admit, it is extraordinarily difficult to answer. The Paleolithic period of our genus Homo spans more than 2 million years, and for much of that period, our climate has been cycling through warm periods and ice ages. And I've drawn a simplified version of these cycles at the top of this slide, showing the approximate intensity and duration of these changes in climate over the past 2 million years. And for about the last 900,000 years, during the late Pleistocene, these cycles have become longer and more intense. And this cycling has had different effects around the world. It has raised and lowered global temperatures. It has changed ocean and air currents. It has joined and split continents. It's made some areas temporarily dry and uninhabitable, other places covered in ice, and other places lush and bountiful. So as we reflect on the period of human evolution, the one thing that has been constant is climatic instability and fluctuation. And during the late Pleistocene, many large animals went extinct. In Australia alone, 88 species of megafauna went extinct. Animals like the giant marsupial lion, the 2-ton giant wombat-like creature, the Diprotodon. In the Americas, more than 38 megafauna genera died out, and they include the woolly mammoth, the giant ground sloth, and the American horse. And humans also likely came to the brink of extinction many times during this period of intense climatic fluctuation. But we managed to survive, unlike our cousins, the Neanderthals. Perhaps it is unsurprising then that what characterizes humans above all else is our versatility. It is this versatility, both biological and cultural, that has allowed us to colonize the globe, even inhabiting the most remote and extreme locations on Earth and space. Since the tail end of the late Pleistocene, during the Upper Paleolithic, the pace of human innovation has dramatically increased. And I'm going to focus on human diets of the past 50,000 years, and especially the past 10,000 years, when human experimentation with domestication turned the human population from 100% foragers to almost 100% agriculturalists and pastoralists. And specifically, I'm going to talk about how our food choices went from this to this in about 500 generations. We've mainly done this in 3 ways. The first is by modifying our own genes in order to be able to digest new types of foods. And the second is by modifying the genes of plants and animals that we want to eat in order to make them more edible for us. And third is by processing our foods using technology to further expand our food options. I'm only going to have time to cover a few highlights, but I think they may include a few surprises. And so let's start with the first one. Let's start with human genetic adaptations. So milk is a food for infants. It contains the sugar lactose, which is actually 2 sugars, glucose and galactose, that are linked by a chemical bond. Now, we can't absorb linked sugars, and so we produce a special enzyme called lactase that acts as a kind of molecular scissors that cuts these 2 sugars apart. Now, all baby mammals produce lactase. But it's not just human babies. Also, kittens and puppies, any mammal really produces lactase, and this is what helps us drink milk as infants. And as part of the weaning process, the gene that produces lactase gets turned off, such that after infancy, after the cessation of lactation, this gene becomes turned off and you no longer produce lactase, and so you can no longer break these chemical bonds. And in fact, if you consume lactose as an adult after your lactase gene has been turned off, this lactase will travel to your colon where bacteria will ferment it, producing gas and acids that cause the symptoms of lactose intolerance. Now, at the end of the late Pleistocene, all humans on Earth were lactose intolerant. Even today, most of the world's population, somewhere between 75 and 90% of people living on Earth, are lactose intolerant. But as you know, some of us can drink milk for our whole lives. Our lactase genes have been permanently switched on because of mutations in this genetic switch. This is a condition called lactase persistence, and these mutations evolved during the Neolithic period. Now, scientists have now mapped the part of the human genome that controls lactase production and this molecular switch that turns it on and off. And we've discovered 5 mutations in human populations around the world that explain almost all cases of lactase persistence. And these mutations trace back to 3 homelands. The first is in Europe and the Middle East, the second is in Saudi Arabia, and there are 3 in East Africa. And each of these places has a long archaeological record of intensive pastoralism of either cattle or camels. Now, with respect to the European variant, it was thought initially that this trait must have evolved with the domestication of cattle about 8,000 years ago, somewhere in Turkey or the Middle East, and then spread with the first farmers into Europe as they traveled in around 7,500 years ago. And this is what all the archaeologists agreed, and we all thought this was a really clever idea, and this must be how it happened. And then in 2007, a really interesting paper was published. This was an ancient DNA investigation of the first farmers in Europe. So this is the linear pottery culture, the LBK culture. And they conducted ancient DNA analysis on these populations to see if they had these genetic markers for lactase persistence. And what they found was they didn't. And then there were subsequent studies because they thought they must really be there. Let's test some more populations. And they kept finding the same result. The earliest farmers in Europe do not have this lactase persistent mutation. And instead, the earliest known evidence for lactase persistence occurs 2,000 years later, and it's found in Sweden and in the Basque country of Spain. And this was quite a surprise. And today among cattle, the greatest diversity in milk-producing genes is found in Scandinavia. This also suggests that this region was really important for establishing modern dairy breeds. And in today, modern Scandinavia has the highest rates of lactase persistence in the world. So more than 80% of people in Scandinavia today have these genetic mutations to allow them to digest milk. And this actually makes sense because life in the far north is quite hard. Food, and especially plants and carbohydrates, are often in very short supply in cold climates. And milk, in addition to being a reliable source of protein and fat and also clean water, is actually a source of sugar. It's a source of lactose. And so by drinking milk in the form of milk itself or yogurt or soft cheeses that still contain lactose, this becomes then a source of sugar that's available year-round. And in some ways, it's like a sugar substitute, or a plant substitute, excuse me. One thing that my research group is currently doing is we're trying to understand the later periods of this. So if these lactase persistence genes start showing up about 5,000 years ago, then when does it start to reach modern levels that we see today? And so we've been analyzing medieval populations in Central Europe to try to understand this period. And try to understand when these alleles stabilized. And what we find is that actually, by 1,000 years ago, we see the same levels of lactase persistence as we do today. And that indicates that this evolution actually occurred extremely rapidly, on the order of a few thousand years. And this is something that I find really fascinating, is we tend to think of evolution over very long time scales, but in some cases it actually occurs quite rapidly. And it's kind of amazing that if you think in Europe that over the span of just a few thousand years, we went from basically no one having lactase persistence alleles to, in some places, more than 80% of people carrying these mutations. That's truly rapid human evolution. And what's even more amazing still is that the most recent evidence from East Africa suggests that the period of evolution there in lactase persistence was even more rapid still. There's a lot of exciting research coming out in this area right now. But this is only part of the dairy story. There's another really important piece of this puzzle that I've barely mentioned. That is microbes, these bacteria that live in your gut. Remember, I said that it's bacterial fermentation in your colon that causes the painful symptoms of lactose intolerance. And theoretically, if you were to drink a half liter of milk and you were completely lactose intolerant and you digested all of that milk completely by colonic fermentation, you would actually produce 8 liters of hydrogen gas, which is terrible. But most people, even people who are lactose intolerant, don't produce anywhere near that amount. And so what's the difference? And it turns out that the gut microbes make a big difference. In fact, there are people who are lactase non-persistent, so they do not have these mutations. They turn off their lactase gene, and yet they don't experience the symptoms of lactose intolerance. So clearly there is something different about their gut microbiome where they aren't producing the acids and they aren't producing the gases and they don't have the symptoms. And this is something that we're quite interested in figuring out. Now, the gut microbiome is extraordinarily complex, and we don't exactly know which bacteria of the thousands of species that live in your colon, which ones are doing this. But there's a lot of work going on right now to try to figure this out, because if we could figure out this process, we could put together maybe a cocktail of some of these bacteria to give to people as a probiotic that would enable them then to be able to digest this milk without all the negative symptoms of lactose intolerance. Another example of very recent human genetic evolution has to do with starch. Now, starch is a polymer of glucose sugars linked up into very long chains. And as with lactose, we can't absorb linked sugars, and so we have to break them up. And to do this, we use a different type of molecular scissors, this time called amylase. Now, all humans produce amylase, and we make it for our entire lives. It's an essential enzyme for life. And on average, we have more copies of the amylase gene and therefore make more amylase enzyme than chimpanzees. And this is part of the evidence that suggests that starch was a really important part of human evolution. But what's also interesting is that some human groups have more amylase gene copies than others. In fact, in 2007, a research team at Arizona State University found that some human populations have many, many more copies of amylase gene than others, and that the number of gene copies actually correlates with the amount of starch in these populations' diets. So, as I mentioned before, chimpanzees just have 1 copy on each chromosome. But they found that in populations that have low starch diets, human groups, they have an average of 5 copies. In other human groups that eat a lot of starch, for example, in Japanese populations, they have on average 7 copies. But they found amazingly that in some individuals, they have up to 15 copies of the amylase gene in their genome. This is truly extraordinary. This is a 15-fold difference in the amount of amylase that they produce. And so in this way, these are 2 examples of how we as humans very recently, on the order of thousands of years, have rapidly developed dietary adaptations to enable us to extract energy out of foods that we either couldn't digest efficiently or couldn't digest at all in the past. Now this brings me now to the topic of domestication. I think, and I hope I'm not too biased, maybe I'm a little biased, but I think domestication is one of humanity's greatest innovations. I think it is so important and so fascinating, but I also think it's quite misunderstood. I think most people are familiar with the fact that we tend to make domesticated plants bigger. So this is an example of wild strawberries. They're actually quite small compared to domesticated strawberries, which are actually a hybrid of a European and a North American wild strawberry. And we do that so there's just more to eat. That's quite simple. And we tend to make domesticated animals smaller, and that's so they're less dangerous and less likely to kill us. But of course, there's a few exceptions, like chickens and guinea pigs, which we also try to make bigger. But domestication is much more than this. In many cases, we have literally invented our own food. We've created foods that didn't even exist before. And a really good example of this is maize, which here in North America we call corn. Now, we're all familiar with corn on the cob, except wild maize doesn't have a cob. That's something we created. Wild maize is that little plant there on the left. It's called teosinte, and I'm going to zoom in on it. Now, it looks nothing like the corn that we're familiar with. It's woody. It has these gloom-covered kernels. They have this odd triangular shape. They're extremely hard. I can assure you, I have tried to eat them. They are very hard to eat. You have to smash them open with a rock. They're not something anyone would have ever been interested in. They're not worth the time of opening. And I think that's what ancient people in Mesoamerica probably would have thought too. Oh, sure, thanks. And so what's really interesting is that if you look at the earliest evidence of maize consumption in Mesoamerica, it's not teosinte kernels that we find. Instead, what we find are bits of stalk that have been chewed up and spit out. And the reason for this is because maize is actually related to sugarcane. It's very sugary. This plant stores a lot of sugar in its stalk. And so you can actually chew on the stalk and suck out the sugars and then spit out the indigestible fibrous parts. And that's exactly what we find in the earliest dry caves. When Mesoamericans first started experimenting with this about 6,000 years ago. But later, there was a mutation that occurred, and it was probably a spontaneous mutation in the teosinte glume architecture gene, which made this glume burst open, and it made the kernel available more readily. Well, this probably is what got people's attention. They started focusing on the seed now rather than the stalk. And then what happens over time is selecting for larger and larger kernels, we eventually get domesticated maize. And this, of course, through artificial selection, allowed people then to build the societies that we're familiar with in ancient Mesoamerica, such as the ancient Olmecs, the Maya, the Mixtecs, the Zapotecs, and the Aztecs. But maize isn't the only food that we've invented. We've actually invented quite a few. Take broccoli, which is another great example. Now, you may not recognize it, but this is wild broccoli here on the left. It's also wild cabbage, wild cauliflower, kale, kohlrabi, and Brussels sprouts. All of these vegetables have been domesticated from the same parent. In fact, they're all the same species. They're just different cultivars. We've just selected the different parts of the same plant that we want to eat. And in the case of broccoli, we have so strongly selected for flowers that you can't even really recognize what it is anymore. This is the same plant, except just lots and lots of flowers all stuck together. And if you don't believe me, I think you should go to the grocery store, and if you put your broccoli into a vase of water, you can actually get it to bloom. And it's quite beautiful. But we've also made new foods in other ways. So when you look out over a wild landscape, and this is a photograph that I took from Gila-Nikit's cave. It's in Oaxaca, Mexico. It's a very early rock shelter site of some of the earliest human occupation in Mexico. People would have lived here about 7,000 years ago, and the landscape would have looked just about the same. And when you look out at this wild landscape, the most immediate problem is not a lack of plants. This landscape is full of plants. But the problem really is that most of them are poisonous or indigestible. And you see, like wild animals, plants are also trying to kill you. But while the animals want to eat you, the plants are just trying to stay alive. They don't want to be your next dinner. And so in order to discourage you from eating them and other pests from eating them, they produce a number of natural pesticides in their roots, stems, leaves, and seeds. Now, we call many of these things flavors, but some of them we also call toxins and poisons. So take ricin, for example. This plant really does not want you to eat it. Ricin is produced by the castor bean, and it's extremely poisonous. 8 beans are enough to be fatal, so watch out for castor beans. Another famous example is, of course, the terribly poisonous nightshade. Every part of this plant is deadly, from its roots to its leaves to its fruits. It contains a cocktail of alkaloid poisons, including atropine, which causes paralysis, and solanine, that's a potent antifungal, and it's also a pesticide poison. What's perhaps less well known is that it's a member of the plant family Solanaceae. That's the same family from which we have domesticated tomatoes, potatoes, eggplants, hot chili peppers, and tobacco. These plants also contain toxic alkaloids and other bioactive compounds, but humans have bred them out of the parts that we like to eat, as in tomatoes and potatoes, or we've refined and concentrated them for a particular effect, as in the case of capsaicin in hot chili peppers, or for nicotine in tobacco. But the poisons are still present in these plants, and that's why you can't eat potato leaves. They still contain very high concentrations of solanine. And this is also the reason why people traditionally peel potatoes. We often think it's to get the dirt off, but that's not it at all. See, if the potatoes get too close to the surface, they'll start making solanine in their skin. You can often tell if this has happened because they also start producing chlorophyll in their skin, which turns them green. So if you see a green spot on your tomato, you shouldn't eat it, not because of the chlorophyll, but because of the solanine that it's producing. But it's not easy to kill yourself on solanine because it tastes really terrible. So you probably wouldn't get that far eating solanine from potatoes. But it can make you quite ill. Tomatoes have a related toxin called tomatine, which they produce. It's a little bit more benign, but it's found in all of the green parts of the plants, especially its leaves. The same principle applies to other plants too. So here we have wild carrot on the left. Some of you may recognize it, like we call it Queensland's lace. And it's really small and woody. It's low in sugar, and it contains high quantities of a pesticide called falcarindiol. But we've bred this mostly out of domesticated carrots. They do have some in the outermost layer of the carrot. That's again why we peel carrots, not to get the dirt off, but to remove this bitter-tasting pesticide. And although you may not realize it, apricots and almonds are actually very closely related. They're both members of the Prunus genus. But in almonds, what we've done is we have bred them to eliminate the cyanide-producing glycosides in their seeds, while in apricots, we have bred them for bigger and thicker fruits. So like broccoli and carrots, domesticated almonds and apricots are fundamentally different from their wild progenitors, and in many ways, a human invention. And we've done this over and over again all throughout the world. We have expanded our food options by either making them bigger or improving the parts that we want to eat, or by genetically altering them to remove their poisons. And this brings me to the final adaptation I'm going to talk about, which is food processing and technology. Now, we've been processing our foods for a very long time. First by using simple chopping and cutting stone tools, and later using much more sophisticated materials and techniques. We've also been cooking our food for a very long time. Hominins have experimented with fire for at least 1.6 million years. And members of the genus Homo have habitually used fire for at least 400,000 years. And in the Middle and Upper Paleolithic, we start finding direct evidence of plant cooking and processing. So for example, starch grain analysis of dental calculus at Shanidara and Spee confirms the importance of plants in Neanderthal diets, including grains, and suggests that some of these plants may have even been cooked or boiled. And starch grain analysis of groundstone tools at Upper Paleolithic sites in Europe has shown that humans were grinding roots and seeds into a kind of flour 20,000 years before the invention of agriculture. Grinding is actually an extraordinarily useful technology, and one that has allowed us to exploit even more food resources. So many roots and nuts, such as manioc and acorns, would be excellent human foods, except for the fact they contain high concentration of cyanide-producing toxins and tannins. But unlike carrots and potatoes, humans didn't breed these toxins out. Instead, we found them useful because it prevented other pests from eating them. And so what we did instead is we developed ways of soaking and leaching out these toxins using technology. So for example, the Canela people of Brazil, they peel and grate their manioc, and then they put them into these long tube baskets and run water through it to leach out these toxins from their manioc plants. And similarly, many native peoples in California produce nutritious acorn flour by grinding the acorns into a fine powder and then leaching out the tannins with water. Cooking and processing, however, isn't just about removing toxins. It can actually improve the nutrition of the food itself. And I think a really good example of this is something called nixtamalization, which is a food processing technique that was invented in ancient Mesoamerica. See, one of the problems with maize is that it actually contains quite a lot of niacin, which is a B vitamin that you require for life. But unfortunately, this B vitamin is in a form that's not bioavailable when you eat it. And so if you try to build a civilization on maize, what happens is you end up with a B vitamin deficiency unless you have a lot of other foods that you can supplement it with. And this is a really big problem. It causes a nutritional disease called pellagra. This is something that really plagued the US prison system for a long time in the 19th century before people figured this out. But what the people in ancient Mesoamerica did was something really clever. One thing they realized was that if you took maize and you boil it, and you add to it something called slaked lime, which is you make that by burning limestone. It's sort of like part of the plaster making process. And if you add that to boiling water and to your corn, what ends up happening is, well, first of all, it loosens the outermost part of the kernel so that it's easier to grind. But it also makes this niacin bioavailable, and suddenly maize has a very large amount of niacin, and you no longer have this B vitamin deficiency. And it also increases the calcium content of your maize 700%. So it dramatically improves the nutritious quality of food. And this particular technique actually got invented over and over again. So in ancient Mesoamerica, they used limestone, called nixtamalization. But in North America, where people were also processing corn, they used wood ashes. Basically, you just need to get an alkaline solution. And so this was hit on over and over again. But food processing also has a dark side. It can also reduce the nutritional value of common foods, and sometimes very severely. So take, for example, wheat. Like other grains, wheat has three main parts, and that's the bran, the endosperm, and the germ. And basically the bran is just the outermost covering of the seed, and it protects it from the environment. The germ is the embryonic part of the plant. This is the part of the plant that's going to sprout. These are the embryonic leaves. And then the endosperm is essentially just starch. And this is what the baby plant uses to feed and grow on until it's large enough to photosynthesize on its own. And during milling, what ends up happening when we prepare flours today out of wheat, we separate these three parts. So we take out all the bran, and we put it in things like animal feed, and we add it to some health breakfast cereals. And then we take the germ out, and we put the germ into jars and sell it at health food stores as a nutritional supplement. And then we take what's left, which is just the starch, and we make it into white flour, and we make rolls and donuts and buns and all sorts of other processed foods out of them. And this is really unfortunate, because if you look at where the vitamins are in wheat, they're almost all in the bran and the germ. So this is where 74% of fiber in wheat is in the bran and the germ. Almost all of the minerals and vitamins are in the bran and the germ. And if you look at what's left when we get to the white flour, it's where most of the carbohydrates, sugars are, most of the sodium. There's not a lot there. We've really stripped out most of the nutrition. And that's why white flour usually has to be enriched with vitamin supplements, because during this process we remove most of the nutritional quality. And you might be wondering, well, why do we do this in the first place? And it really comes down to two things. One is aesthetics. It makes a lighter colored flour if you remove the bran and the germ. But also it increases shelf life, because the bran and the germ are more bioactive, they contain more oils, and the flour will spoil more quickly if you leave it in. But if you're more interested in nutrition than you are in aesthetics or shelf life, then what you can do is look for whole grain wheat, because that contains the bran, the germ, and the endosperm together in their natural proportions, and you get the full nutritional benefit of the wheat. But perhaps the most alarming trend in modern food processing today is the extreme concentration of certain nutrients, especially sugars. So the most common and perhaps most harmful example is soda, which is actually really a kind of liquid candy. Soda is really just this absurd concentration of a natural product that is almost always rare in nature, and that's sugar. We're not evolved to eat large quantities of sugar. In fact, before the 18th century, few people had ever even tasted refined sugar. Now, fruits and honey contain some forms of sugar, but they also contain a lot of other things. They contain fiber and pectins and complex oligosaccharides, and they aren't really comparable to the kinds of refined sugars that you see in soda. Also, the quantity of sugar in soda is really jaw-dropping. So, for example, if you take this lovely 20-ounce Sunkist, it contains 72 grams of sugar. which is really astounding. So 72 grams, that's 36 sugar packets. So I just want you to imagine in your mind, you're preparing coffee in the morning, you're just putting in 36 sugar packets. It's really quite absurd. And like, what's even more crazy is if you had to go, if you were an ancient person and you had to go acquire this much sugar from a natural source, well, you'll have to work really hard because this is how much sugar cane you would have to eat in order to consume 72 grams of sugar. It would almost be physically impossible for you to do it. I mean, it would take some serious effort. And at least in Oklahoma, where I live, what I see is actually not that many people drinking sodas of this size. I see a lot of people with 44-ounce sodas, which is just crazy. And that's about, let's see, well, this is a 6 feet of sugar cane. That's about 91 packets of sugar, which is just crazy. You're almost baking a cake at that point. So just keep in mind how much sugar is in it. The real problem is, you see, is that we can't modulate our digestion of sugar. I mean, this was something we never had in abundance in the past, so we don't really have any defenses to sugar. We digest it immediately, and it causes extreme spikes in our blood sugar, and it wreaks havoc on our pancreas and our long-term metabolism. So from biological and behavioral ingenuity to the perils of plenty, this is how we as humans have eaten our way out of wild landscapes and into a modern supermarket. And for the first time in human history, starvation is no longer the most pressing concern for most people on Earth. As the threat of obesity and metabolic disease looms in the 21st century, we must again be adaptive, and we must change our current economic systems and food processing technologies to improve our nutrition rather than to erode it. So, thank you very much.